Welcome to one of the scariest nights of the year, Halloween. But tonight's spooky story isn't about ghosts. It's about something truly shocking: genetic mutations. Now introducing one of the country's best genetic mutation experts ever.
Hey, I'm Dr. Hoof, a specialist in genetic mutations and the strange ways they shape the animal kingdom. So, a mutation is a change in an organism's DNA. Sometimes, mutations do nothing, but other times, they can cause changes in how the body works or how it reacts. Today, we'll be looking at one that turns fear into something unbelievable.
Meet Billy. Unlike other goats, Billy has a mutation called Myotonia congenita, a mutation that affects his muscle in an unordinary way. When Billy gets startled, let's just say he does not run. Yep, that's right. Billy doesn't just flinch, his whole body freezes up.
This happens because of a mutation in the CLCN1 gene. Normally, this gene helps produce a protein called the chloride channel. This protein regulates the flow of chloride ions in the muscle cells, allowing muscles to relax after contracting. But, in Billy's case, this mutation disrupts the chloride channel. The chloride ions get stuck, and his muscles stay locked in a place when he gets startled.
So, instead of running away from danger like other goats, Billy's muscles freeze up, making him an easy target for scares.
Now, I think it's extremely important to mention that this is completely harmless to Billy and other goats. This mutation won't affect their health or cause injury, as it just temporarily freezes their muscles. So, even though it looks dramatic, we promise Billy's body is completely fine after he gets up. So what happens when Billy enters the scariest haunted house on the farm? Let's find out.
I can't do this. It's too scary for me. My doctor says I should really be going outside, but I've heard going out at night is really bad for your body, you know? It's like unhealthy. And the air is really cold out here, and I'm like pretty sure I'm allergic to dust or something. And who knows what's lurking in there? It's just spooky, right?
Quit stalling, Billy. You've been standing here for ages. It's just a haunted house, not a death trap.
Oh no. What is that? What is that?
Billy, we haven't even started the haunted house.
And there it is. Billy is frozen once again, thanks to a little thing called Myotonia congenita. His body locked up the moment he got startled. This time, it was a simple cobweb, but the result is still the same: a dramatic fall.
You see, everything in Billy's body, including how his muscles work, is controlled by the instructions written in his genes. But, for those instructions to be followed, they first have to be copied through a process called transcription, where the information in your DNA is used to create messenger RNA, or mRNA. It's crucial because without it, proteins can't be made, and without proteins, nothing would function in your body. Before we dive into how that happens, let's talk about the building blocks that make the process work. Nitrogenous bases. DNA is made up of 4 bases: adenine, thymine, cytosine, and guanine. These bases pair up in a specific way. A always pairs with T, C always pairs with G. That's simple. But RNA is a little different. Instead of T, RNA has U for uracil, so A pairs with U in RNA. This change is important because it allows RNA to function separately from DNA while still carrying the same genetic message. DNA is like a blueprint. It stays safely in the nucleus because if it leaves, the cell might think it's a virus and attack it. But the cell needs to build proteins outside of the nucleus, in the cytoplasm. That's where RNA comes in messenger, or mRNA form, as it acts like a courier, carrying the genetic instructions from the DNA in the nucleus to the ribosomes, where proteins are made. The first step is prepping the DNA, but how exactly do we do that? The enzyme helicase separates 2 strands of DNA by unwinding its double helix. This allows the information to be read. Picture it like this. You've got a twisted up rope, and helicase is the hand pulling it apart so we can access individual threads. Now, once the DNA is open, we are ready for the main enzyme in transcription, RNA polymerase. This enzyme is like a builder. Once RNA polymerase is in position, it begins reading one strand of the DNA, called the template strand, and makes a complementary copy of it in RNA form. Many might ask me, well, Dr. Hoof, why does RNA polymerase make a copy of RNA instead of just copying DNA? To put it simply, RNA and DNA are different. It's like a close cousin, but it has a few differences. The key difference is that instead of thymine, RNA has U, or uracil. So, when RNA polymerase reads an A in the DNA, it adds an U to the RNA strand. In C, it pairs with G, and for G, it pairs back with C. This process is called complementary base pairing. So, complementary base pairing is kind of like a puzzle piece, as you have to fit them all together to make sure the message stays accurate. As RNA polymerase moves along the DNA, it builds the mRNA strand by adding the correct complementary bases, and soon we have a full mRNA molecule. The molecule carries the genetic message from the DNA to the rest of the cell, but it needs to leave the nucleus first, because it can't stay in the DNA. It's like sending a message out of a locked vault. RNA is the messenger that takes the information where it's needed. The reason we use RNA instead of DNA for this process is simply because it's much more flexible. It can leave the nucleus and travel to the cytoplasm, where it's needed to make proteins. This is where Billy's mutation is affected: transcription. Transcription is responsible for the CLCN1 gene properly transcribed to create the correct protein. But, as we know, this mutation messes up this process, resulting in faulty mRNA. Speaking of Billy, I totally forgot about this whole dilemma because of how awesome transcription is. I wonder how he's doing. Let's check in on our favorite goat. Billy, Billy, Billy! Can you get up already? You've been still for what seems like forever. Okay, we can leave Billy for now. He seems fine to me. And we have some unfinished work to do, specifically translation. Okay, so now that we have our mRNA ready, we're on to the next major step: translation. This is where we take the message in the mRNA and turn it into a protein. But how does that come about? Let me explain. The first codon in translation is always AUG, also known as the start codon. This signals the ribosome to begin translation and also codes for the amino acid methionine. So, the ribosome calls in a different type of RNA called tRNA to deliver methionine. tRNA, or transfer RNA, is like a delivery truck. It carries amino acids to the ribosome. Each tRNA has an anticodon, a set of 3 bases that are complementary to the mRNA codon. The first tRNA has the anticodon UAC, which perfectly matches with the AUG on the mRNA. As the ribosome moves along the mRNA, it reads the next codon. Let's say the next codon is CCG. The ribosome looks for the tRNA with the matching anticodon, which would be GGC, and this carries the amino acid glycine, or GLY for short. Once the tRNA delivers the amino acid, the ribosome forms a bond between methionine and glycine. This bond is called a peptide bond, and it's what connects amino acids together to later on form a protein. This process continues until the ribosome reaches a stop codon, either UAA, UAG, or UGA. These codons don't code for an amino acid. Instead, they signal that the translation is complete. The ribosome will then release the polypeptide chain to get folded and edited in the rough ER. Now that we understand transcription and translation, let's talk about what happens when something goes wrong. For this case, Billy's myotonia congenita. Billy's mutation is caused by a missense, which means that a single base substitution changes one amino acid in the protein. Let's say the normal DNA sequence in the CLCN1 gene contains GGC, which is transcribed to CCG in mRNA and codes for proline. If a point mutation makes it CUG, then it codes for arginine, not proline. This can totally disrupt the shape and function of the gene, preventing it from working. Well, that's all I'll say for now. Hope you understood the science behind this wacky mutation.
Yo, Bruno, what's that?
What's what?